So it's been a while since we've heard from you because you've been really busy. And based on the video I saw on YouTube from Horniman featuring you, um, you're a new daddy, right? To a new brood of Acropora spawn.
Yeah. So November and December were really busy for the team. We had quite a big spawn in November. Probably, I think there was about 30 colonies went in November, and then December we probably had about another 40 colonies spawn then. So in total, about 300,000 eggs were released, and they were from 5 species. We ran a few experiments, so some of it was looking at hybridization. Others, we were preserving samples, looking at the embryogenesis stages. So the stages from fertilization right the way through this sort of free-swimming larvae stage. So they're going to be run on, well, I've been doing some electron microscopy imaging. Now we're looking at something called confocal laser scanning microscopy.
Hold on. Explain in layman's terms what those two different types of microscopy are.
Okay. They both use electrons. So the scanning electron microscope basically allows you to look at things in incredibly fine detail. But you're looking at the surface structure. You can't look down through the tissue. If it's not opaque, you can't see through it. So what we've done so far is imaged all of the different stages, the cellular stages of, or the cellular division, going right the way through the 4-day process to get the free-swimming larvae. So the way the corals spawn, they're releasing eggs and sperm, fertilizations are occurring up at the surface of the water. Once fertilization happens, you start having cell division, and then over a course of about 4 days, this free-swimming larvae forms. And at that point, this is all happening up at the ocean surface. Once you have this free-swimming larvae, it's basically dropping down, settling on the reef, and growing into the primary polyp. So that's the process. So the electron microscopy was just imaging those different stages. So we took samples at set periods through that 4 days, and then mapped out that embryogenesis. But we're looking at the surface structure. So laser scanning, a confocal laser scanning, what we are able to do, it looks at fluorescence. So you can apply different fluorescence to different structures in the cell, depending on what you want to look at. So what I'm looking at doing next week with some of these preserved samples, I've got a fluorescent dye that will stick to DNA, and it will turn the DNA blue. So what that'll do is mark the nuclei in the different cells. And I want to look at the relationship between the way the sperm enters the egg, because I think there's something called polarity of the egg. So the egg is perfectly round. I think the nucleus is actually residing in one hemisphere, and the sperm are attracted to that hemisphere. But with electron microscopy, we can only see the outside. So that's why I think this is happening. With this confocal, we'll mark up with a fluorescent dye, and then basically fire different wavelengths of lasers in exactly the same as the way the radions work, right? So different lasers and different peaks and troughs within the spectrum, and that will basically highlight the nuclei, and then we can look at that relationship. But what this technique allows you to do is then look down through the structure and create 3D images, so you can really understand where these
And what you're interested in looking at here is if we imagine the egg as a sphere and the nuclei of the egg being the genetic code center, is, you think, not directly in the middle, but actually near one of the poles, which is, because it's a sphere, magnetic, so the sperm knows where on the egg it's targeting when it what's the word for inception? Or
oh yeah, exactly that. So the polarity, that's exactly right. I think there's the egg is divided into 2 poles, what's called the animal pole and the vegetal pole. The animal pole is where cellular division's happening quickly, whereas the vegetal pole is where lots of fats and some of the sort of energy of the egg is residing in that lower section. But cell division happens much slower in that vegetal.
So what's the significance? Is this just for better understanding, or will this help you increase your fertilization rate somehow? Or
the significance is simply documenting. This is an unknown at the moment, and so a lot of that embryogenesis, there's no significance in terms of increasing fertilization success. It's simply understanding the process well enough. Yeah.
Very neat. Very cool. Awesome. Well, thank you very much, Jamie, and we'll catch you next time.
Talk soon, yeah. Cheers.
Cheers.
Hopefully without fire off. Yeah. Take it easy.